Hello friends, I am Dr. Shaha. I am a molecular biologist with expertise in microbiology, genetics, and bioinformatics. On this channel, I break down complex biological concepts into easy to understand content. If you're passionate about science, subscribe and join me on this journey of discovery. Hello everyone, welcome back to my channel. Today I have come with a new sort of video. So I will discuss the top 10 research topics in molecular biology pretty much in detail. So it will actually help you designing your project and ultimately doing the research actually you like to do. All right, let's dive in. The first research topic that are most popular currently in molecular biology, I would say CRISPR and gene editing. CRISPR-Cas9 has revolutionized genetic engineering, allowing precise gene modifications in various organisms. Applications of this technique are actually currently under different phases of trials, but most likely the recent advance in this technology, we can say probably treating genetic disorders like one example I can say sickle cell anemia or cystic fibrosis. The other application includes developing genetically modified crops with improved traits, potential ethical concerns, especially regarding human germline editing. So that's actually probably come with this technology, but scientists are currently doing great research on that and to make this technology accessible and how safe it would be. They're working on it. And some trends which are actually emerging behind these CRISPR and gene editing could be base editing and prime editing for even more precise DNA modifications. And this technology is currently providing a lot of good information and new techniques, how we can actually do most advanced biological activity, not only in different plants or trees or bacteria, but also in some animal models. But these are actually currently ongoing, so probably have to wait a bit to hear more of these technique in detail. But that's really a good topic currently to do research in molecular biology. The second one I could say like epigenetics and gene regulation. So epigenetic studies means that how gene expression is altered without changing the DNA sequence, often through DNA methylation or histone modifications and non-coding RNAs. So the applications are currently versatile for this technique. So the current applications of this epigenetic and gene regulation technique could be understanding cancer progression and resistance. So as you know that there's no any updated treatment for cancer except the traditional one, chemo radiotherapies, but the epigenetic research is currently providing much more information of how a cancer can be prevented and how can we understand what sort of personalized treatment could be given for cancer patients. So these are currently ongoing. The third one I can discuss about single cell genomics and transcriptomics. So traditional bulk sequencing averages gene expression across many cells, whereas single cell technologies allows analysis at the individual cell level. So there are applications for these single cell genomics and transcriptomics area. So some are understanding tumor heterogeneity and drug resistance in cancer. So it is not really uncommon to see the treatment or the chemotherapy is not working for a particular cancer type. So, what could be the reason behind it, and how we can overcome these resistance that probably could be a part of single cell genomics and transcriptomics analysis and of these area. Studying early embryonic development and stem cell differentiation. So, as you know, when the embryonic development happens, so there are some ways that we can actually modify the trait. And when the stem cell differentiate into multiple different set of cells inside the body, there are some chances probably that these cells could be modified to gain a better characteristics. That could be done by single cell genomics or transcriptomics methods. And the other thing we can think about this area could be mapping neural circuits in the brain. So we know that the brain functions are really complex, and there are a lot of neural activity, and there are a lot of different sort of mapping systems happens in the brain. So the brain biologist could say more about that. But currently, this technique is really versatile, and we can use these single cell genomics and transcriptomics techniques to understand a lot of in-depth biological questions that currently the scientists are going on doing research on. So key technologies of these area could be single cell RNA sequencing, which is called RNA-seq, and the ATSC sequencing or spatial transcriptomics. These technologies could be used in these areas, single cell genomics and transcriptomics, so that we can actually discover much in-depth information about molecular biology. The number 4 could be synthetic biology and genetic circuit engineering. So the design and constructions of biological parts and system to create artificial cells, biosensors, and therapeutic molecules could be the underlying part of the synthetic biology and genetic engineering study. So there are currently potential applications for these area. So, for example, engineering bacteria to produce biofuels or clean up environmental pollutants, or generating a specific protein which could be used for medical treatment or disease prevention. So another application could be developing synthetic probiotics to treat gut disorders. As you know that the probiotics are really playing a major role in this area to prevent some of the disease, or to even to cure some of the disease as well, which are mediated by different pathogenic bacteria. So these synthetic biology and genetic circuit engineering could be beneficial for this purpose. Another applications of this technique could be programming cells to act as biosensor for disease detection. So as you know that in some of the cases, the diseases are not detected primarily, and it's only detected when actually you have very less thing to do. And this technology could actually program individual cells to act as a sensor, so that it can say very early time that there is something happening we should be taking care of. That's for the synthetic biology things. And number 5, I can say microbiome research and host microbe interactions. So this is really a specific interesting part of biological, basically molecular biology research. And the microbiome research is currently getting too much attention on that. There are reasons for that. And that's really brings a lot of good things. So the human microbiome plays a crucial role in metabolism, immunity, and disease development. There are some applications of this microbiome research. For example, the microbiome-based therapies for different diseases, including obesity, diabetes, and depression. So these are actually found correlated with the gut microbiome. There are several research on it, published papers on it. So these definitely could be one of the interesting topic to do research on molecular biology. So there are other applications for this microbiome research. For instance, fecal microbiota transplant, which is called FMT, to treat antibiotic resistance infections. So currently, as you know, that we are actually living in the area where people are getting a lot of antibiotic resistance bugs in their bodies. So the bugs are actually also getting resistant to different conventional and the advanced antibiotics currently available. So these transplant, fecal microbiota transplant to actually give the antibiotic sensitive microbiota and then taking the resistant one could be one of the research area for microbiome and host microbe interaction research. So there are some future direction of this research like personalized microbiome therapeutics and probiotic engineering. So if you can actually make the microbiome personalized for an individual, and depending on actually what sort of immunity and microbiome they got. So that could be one of the standout research in this area, I would say. All right, so let's go to number 6. Number 6 is RNA biology and therapeutics. So RNA is not just a messenger. It plays crucial regulatory roles in the cells. There are applications of RNA biology like development of mRNA vaccines. As you already know that COVID-19 vaccines by different companies actually were the RNA vaccines. And these, until now, these vaccines are really of good success. And there are other applications for RNA biology like RNA interference, therapies for genetic engineering. So these are also the ongoing research area of molecular biology. And some of the RNA biology research also cross-connected with the CRISPR method. So CRISPR-based RNA editing techniques, for example, repair, rescue, these tools are currently available for doing RNA research, which are also engaged with CRISPR technology. And there are some key areas of this RNA biology and therapeutic research. For instance, long non-coding RNAs. These are really a new sort of RNAs discovered and are going to be really beneficial for doing further RNA research. And circular RNAs and RNA modifications. So RNA modifications could be something like M6A methylations, which is really targeted currently for different diseases. So these RNA biology research are really outstanding. So we can move forward to number 7, which is protein folding and misfolding in disease. So misfolded proteins are implicated in neurodegenerative diseases like Alzheimer's, Parkinson's, or Huntington's disease. And some of the applications of protein folding and misfolding in disease could be development of molecular chaperone-based therapies, understanding prion-like spreading mechanisms in neurodegenerations. So these diseases are actually considered to be really advanced sort of disease, which are actually the results of protein denaturations or misfolding things. So the proteins are not in the natural circumstances. It changes. That's why these diseases are actually happening. So there are a lot of spaces to do research in this area and to give much more information how we can actually control these diseases. Other applications of these protein folding and misfolding could be targeting protein aggregation in small molecules or gene therapies. So there are some emerging approaches as well for this area. AI-driven protein structure prediction. So we know that the protein folding and misfolding are associated with protein structure. So if the structure is natural, then the protein probably functions naturally. But if the folding are actually impacted by several other regions, then the structure is also going to be changed or disordered. So that's why the AI can help predicting the protein structure, the correct protein structure, and then probably we can work on how we can do this in the laboratory. All right. So let's go to the next one. Number 8 is systems biology and computational modeling. So currently the bioinformatic research are really prevailing over anything. And there are reasons for that. The bioinformatic research require less funding and more outcome. So that's why this technique is now very popular. So how the system biology works? It integrates genomics, proteomics, and metabolomics data to model biological networks and disease pathways. So that actually tells specifically which pathway or which network is disrupted for a disease. So we can work on this area particularly. So there are some applications currently available for these system biology approaches, like identifying novel drug targets using in silico simulations, studying metabolic reprogramming in cancer and bacterial or viral infections, modeling host pathogen interactions for vaccine development. So these broad applications are really facilitating, I guess, for the system biology research. And there are much more actually to give in this area as well. And this area is just widespreading. So we hope that very soon we'll be getting much more advanced technique in bioinformatics so that we can study not only how disease causes, but also how disease can be prevented. So there are key techniques for doing these research, system biology research, like machine learning, network biology, and multiomics data integration, as we discussed earlier. So let's go to number 9. It's molecular mechanism of aging and longevity. So as you know that aging is driven by molecular changes such as DNA damage, telomere shortening, and mitochondrial dysfunction. Applications of these technique could be anti-aging interventions like senolytics. There are a lot of information coming out currently about senolytics and how we can use these as an anti-aging method. So, but research needed for that area. Investigating the role of sirtuins and autophagy in lifespan extension. So this could be a part of these molecular mechanisms as well. If we know in which molecular signaling or checkpoints are working for these aging related things, then probably it will actually change the world. Another application of this technique could be studying long-lived species. For example, naked mole rats for longevity insights. We can actually see how their lives are actually working, how they're living long, and then probably we can get some hypothesis and we can try these in different ways. So there are some emerging trends of these molecular mechanisms of aging research, like epigenetic clocks to measure biological aging and rejuvenation therapies. So that's it for molecular mechanisms of aging and longevity. So we are now at the end. So I'll be telling you the number 10th research. So this is actually cancer genomics and precision medicine. So understanding genetic mutations and epigenetic changes that drive cancer progression. So this is particularly important because it's a long time we're doing research in cancer to find out a way, a best way to actually prevent cancer progression or cancer cure, but we're still on the way. That's why the cancer genomics is pretty much new area we can actually work on, and we can find out ways how to defeat cancers. There are some current applications of these cancer genomics and precision medicines research, like developing targeted therapies, for example, CAR T cell therapy, immunotherapy, and identifying biomarkers for early cancer detection. So this is also an important area because if we can diagnose cancer early, there are chances that we can actually cure this, probably. But most of the cancers actually are detected in late stage when there is very less thing to do. So we really want to identify some biomarker for early cancer detection. And other application of this technique could be using liquid biopsies, like circulating tumor DNA for non-invasive monitoring. So this is probably one of the research area which is pretty much neglected, I can say. So we can actually prioritize this area to understand how cancer invasion happens and where is the specific checkpoints that we can actually think about to prevent the spreading of cancer, right? So there are some key projects under these category like Cancer Genome Atlas. They are actually working really good to make a platform of cancer data, cancer cell data, and single cell sequencing in tumors. So we can actually also think about doing single cell sequencing to understand how the tumors transcriptomes and proteomes actually works, and some AI-based cancer diagnosis. So we can generate AI tool by which from specific informations we can predict what cancer and what stages of the cancer currently at this moment. So these are really the innovative ways we can put our research on. And that's it. Would you like me to expand any of these topics in future? Just let me know. I'm pretty much open to that. And also leave any of your comments or suggestions in the video comment, and please subscribe to the channel. So I'll be coming with a new videos and tools of your likes. So in future, stay with us. Thank you so much. Bye.